in the interest of time, just going to give a few center updates. Thank you all for joining these monthly lectures, and they're always very nicely attended, and we all learn a great deal. But a couple of Dunlevie Center updates. We announced our Spring Seed Grant awardees. It was a competitive field, but I am delighted to announce that Dr. Pasca and Dr. Bianco, their projects were selected and are being funded. So congratulations to both of them, and also a warm welcome to Dr. Matteo Moli, who is joining us from the United Kingdom, and will be faculty assistant professor in the perinatal biology section with Virginia, and working in the Dunlevie Center Basic Science Discovery domain. We're delighted to have you here, Matteo, and look forward to all the good work that you'll be doing. And with that, I will, oh, yes, one more, sorry. We have our next speaker is going to be Anushka Tseshewitz, who is an assistant professor of pediatrics and stem cell transplantation, and will be talking about a project that we are unfolding on in utero transplantation for Fanconi anemia. It's a very exciting development. And I believe that is the last intro slide, correct, Janina? Wonderful. Okay, and I will hand this over to Dr. Virginia Nguyen.
Great, thank you, Yasser. It is my great pleasure to introduce my colleague and friend, Dr. Emily Su, who is a professor of Obstetrics and Gynecology at the University of Colorado School of Medicine. As many of you know, that's where I came from when I came to Stanford, and we barely overlapped at Colorado. But Emily did most of her medical training, fellowship training in MFM, as well as a Master's of Science of Clinical Investigation at Northwestern University in Chicago. She has been at Colorado and has climbed through the ranks beautifully, from assistant all the way up to full professor. Key highlights in regards to recognition of the impactful research that she has done. She's received the SRI President's Achievement Award. She's received the International Federation of Placenta Associations Career Research Award. She was honored as Mentor of the Year for District 8 of ACOG. And most recently, she has been inducted into the American Gynecologic and Obstetrical Society, or AGOS, which is a tremendous honor. I think it's always fun to see where people's scientific careers started. So Emily's first published paper was on brain research, where she studied evidence for studies with ethylmalamide and 12-O-tetradecanol 4-ball-13 acetate, that AP1 and CREB are involved in the glucocorticoid activation of the TRH gene expression in hypothalamic cultures. Since that time, steroids is a common theme of her research career. But her most recent work is really understanding endothelial health and endothelial cell biology and angiogenesis in the placenta of fetal growth restricted pregnancies. And I'm so excited to see what she has to share with us today. Emily, it's all yours.
Great, thanks. Can you guys see my slides okay?
Yes, yeah. Perfect.
Hold on one second. Let me just move the little screen of talking thing over here. Thank you so much for the introduction, Virginia. It's great to see some familiar faces, even if it's via Zoom. And, you know, it's so funny because what you just described as my first publication. One, I can't tell you what it's about. Two, I don't think I can pronounce any of those words either. And three, it just goes to show you that I never really was planning on any sort of a basic science career. So yeah, my job description is definitely very different than what I had anticipated. So as Virginia had said, my main research interest has revolved primarily around severe early onset fetal growth restriction with absent or reversed umbilical artery Dopplers. And I know that's a super specific pathologic phenotype, but this particular topic sparked my interest a long time ago during residency. And so I've had the privilege of taking care of some wonderful individuals whose pregnancies were complicated by severe growth restriction. And these are just a few photos that they've allowed me to share. And while several of them I think are quite happy, it not only took months of intensive neonatal care and long-term follow-up to get to these points, but many other families weren't so lucky and unfortunately experienced stillbirth, neonatal death, or more severe long-term complications as a result of prematurity that were further compounded by severe growth restriction. And again, you know, I never anticipated having such a research-intensive job description, much less one that's so focused heavily on basic science. Whatever Virginia described, I remember in college, kind of like checking off tick boxes of things I needed to do, but really kind of wanting to poke my eyes out doing the project. But I think in residency, suddenly seeing the impact of this diagnosis on families and how there was so little we could really do showed me how far behind we are in obstetrics when it comes to understanding mechanisms of both normal and pathologic processes. And so as just one example, my dad passed away from AML, which is a diagnosis that about 300,000 people receive worldwide each year. And at the time of his diagnosis, about 10 years ago, his 5-year survival rate was 10%. And unfortunately, I say his body read the statistics and he died about 1 year later. But in the decades since his diagnosis, there have been such substantial advances in understanding the mechanisms of this disease and in development of therapeutic interventions that now the 5-year survival rate for his type of AML is 90% instead of a very measly 10%. In contrast, 140 million women give birth worldwide each year, and only 300,000 people worldwide get acute myeloid leukemia. And yet we still don't have any idea why humans go into labor. And it boggles my mind that we still don't understand mechanisms underlying something that is so universal as parturition. And I know I'm preaching to the choir, but this is just one example, I think, of why we need to continue to push the boundaries of obstetric research. And so I'm not going to go into the definition of severe early onset growth restriction in this to this audience, but we're primarily interested in those specifically with absent or reversed umbilical artery Doppler velocimetry. And so among growth restricted pregnancies, as you guys know, fetal placental Dopplers are most predictive of adverse perinatal outcome. And more specifically, they reflect the status of placental vascular resistance. And obviously, as this audience knows, the worse resistance you get, the higher your pregnancy is at risk for fetal hypoxia or death. I think in the absence of delivery. The problem, though, is that despite our ability to predict adverse perinatal outcome, clinical data show that besides antenatal steroids and maybe MAG for neuroprotection, our current obstetric management paradigms really aren't meaningfully impacting outcomes. And so, again, this audience knows this, but what I mean by this is that our overall goal is to try and time delivery where we're maximizing gestational age while minimizing prolonged exposure to an abnormal in utero environment. But the reality, I think, is that these fetuses, by the time they get this diagnosis, that they're so far compromised that we're really kind of stuck between a rock and a hard place in terms of management. And so on the one hand, if we try and cut our losses and deliver to get or on the one hand, if we try and maximize gestational age and decrease the risks of prematurity, we get more stillbirths. If we cut our losses, on the other hand, and minimize prolonged fetal exposure to a hypoxemic environment, we get fewer stillbirths, but we trade those for neonatal deaths. And then, as you guys know, because of a substantial preterm delivery, a substantial proportion of these infants who survive beyond the neonatal period will go on to have long-term complications secondary to prematurity that's worsened by their growth restriction. And then in terms of other interventions besides just delivery, I think we'd agree that data suggests no consistent efficacy with aspirin, heparin, or even sildenafil treatment. And so although we have tools, I guess, they're just not necessarily the right ones for this situation. And so our lab is hoping that understanding why placentas develop this degree of insufficiency can start to move the needle in terms of clinical care. And so from here on out, I abbreviate severe growth restriction with absent or umbilical artery Dopplers as FGR-ADV, just to distinguish it from run-of-the-mill growth restriction. And so one main reason placental vascular resistance is abnormally elevated is that these placentas have a smaller fetal placental vasculature, which can be seen by experimental 3D Doppler images. From stereological depictions of what's going on microscopically in the terminal villi, we can see that these vessels and villi from these severely growth restricted pregnancies have these abnormally thin, poorly branched vessels in contrast to villous vessels that are normally branched. And so this impairment in angiogenesis leads to diminished villous vascular volume, which then structurally contributes to this increase in vascular resistance that we then see clinically by our Dopplers. And pathophysiologically, this decrease in villous and vascular surface area impairs oxygen extraction from the maternal intervillous space. This results in fetal hypoxemia or hypoxia, and then add on to that a fetus that has to pump extra hard against this high resistance placental vasculature, and you get deterioration of fetal cardiac function, and then stillbirth if delivery doesn't occur expeditiously. And so our lab—
Can I ask a question?
Sure.
Thinking through that pathway. So the worsening of Dopplers, do you think it's actually you're getting simplified vasculature and simplified branching villi, i.e. they were better branched, but with time they're getting less branched, leading to increased vasculature resistance?
That's a great question. I don't know that anyone knows. So all of like the classic placental pathology, like the Benershka texts, and then like John Kingdom's work at the University of Toronto suggests that these vessels are poorly branched. And because I think we think of angiogenesis as normally ongoing throughout all of pregnancy, and because this branching angiogenesis should really occur between 25 weeks to 40 weeks or term or whatever you want to call it, that I think that's why it's been shown that it's been considered to be more of a deficiency in angiogenesis. I think it's possible, and actually even the next image sort of alludes to it. I actually always wonder if it's possible that cells are sort of moving and having displaying some sort of angiogenic phenotype, but then they can't stabilize their vessels. That's something that we're looking into, but really because we've shown primarily migrational defects in these cells that we isolate, which I'll show here. We've focused more on kind of mechanisms leading to deficient migratory proliferative kind of properties. Does that sort of answer your question?
Yeah,
I think it's worth keeping in mind, definitely, that it might be a vessel stabilization issue.
Okay, great, thank you.
So as I had just said, our lab isolates fetal placental endothelial cells from these severely growth restricted pregnancies. And we also take cells from gestational age matched appropriately grown preterm controls, and then from full term controls as well. Pros and cons to both of these control groups, obviously, which is why we have two. But these cells that are isolated from these growth restricted placentas exhibit deficient migration as compared to these gestational age matched appropriately grown controls. And so you can see that by tube formation assays, these growth restricted cells, show fewer branch points and less total length. Again, this is something that one of the things we could do, I think, is to look at this under live cell microscopy and see if the tubes are forming and then they're falling apart, as Virginia alluded to, or that they're just not moving that well in general. And actually from some live cell imaging otherwise, since then, on a more like wound scratch kind of analysis, it looks like there definitely is some sort of migratory abnormality. And so to get a more detailed understanding of what was going on a cellular level, again, we did these wound scratch assays where these cells are plated to confluence and you make a scratch, and then cells should move into the direction of the wound in order to close it. And you can see that in control endothelial cells, cells are extending and elongating towards the direction of the wound in order to close it. Whereas in growth restricted cells, the polarity of the actin is actually parallel to the wound with minimal, if any, filopodia and lamellopodia. And so this data suggests that there are inherent angiogenic defects in growth restricted cells, but there have been two main things that have continued to niggle at me. So first, the wound scratch figure on the right clearly shows endothelial cells that are cultured on tissue culture plates, which is not representative on so many different levels of what's actually happening in vivo. And the other issue is that even in the setting of matrigel for the tube formation assays, which is an extracellular matrix that promotes angiogenesis, growth-restricted cell migration still wasn't able to be rescued, which was making us wonder whether this could totally be explained by just inherent problems in the growth-restricted cells alone, or whether there would be different responses to tissue-specific matrix that better recapitulates the in vivo placenta. And so we took a cue from tumor angiogenesis literature. And one aspect that really resonated is that not only is the tumor microenvironment a critical determinant of angiogenesis, but the surrounding stroma is a key regulator of focal adhesions and actin cytoskeleton dynamics, both of which are key mechanisms that you need in order to get endothelial cell, proper endothelial cell migration. And at least on tissue culture plates, they look like there might be some qualitative differences. And so the most basic parallel of the tumor microenvironment in the placenta is placental villostroma, which surrounds the fetal placental vasculature. And of all the cell types present in the stroma, placental fibroblasts are the most abundant and the main cell type that secrete extracellular matrix. And so within a pre-existing blood vessel, most commonly the microvasculature, like fetal placental capillaries, an endothelial tip cell is designated, and then somehow through secretion of matrix metalloproteases or other enzymes, the basement membrane undergoes degradation, and then the tip cell starts to move. And then several parameters control this sprouting angiogenesis, including sequestration of soluble angiogenic factors like VEGF, adhesion cues through extracellular matrix proteins like fibronectin, and then the stiffness and rigidity of the matrix or the stroma itself is also super important for endothelial cell angiogenesis. And so with proper interactions between the cell and the matrix, these endothelial cells proliferate and migrate. And then this image doesn't show it, but these vessels need to stabilize too. And then you get the end result of hopefully normal angiogenesis. And so to date, what we know about placental stroma is really limited. There, about 2 decades ago, someone data mined an existing human mRNA database for existing genes, and that showed that fibronectin and collagens were the most abundantly expressed ECM proteins in the placenta. There have been several small immunohistochemistry studies that are older as well, but I think my understanding is that depending on how well the tissue was fixed, matrix proteins itself on like FFPE slides can either have a lot of non-specific staining or poor antibody binding because of the way those samples were prepped. So those studies are a little bit more limited. More recently, there have been shear wave elastography papers, and one group of 2 or 3 years ago combined all the data together, and they found that there was altered placental stiffness with pregnancy pathology, with growth-restricted placentas exhibiting nearly a 10-fold increase in stiffness as compared to normal placentas. And so to better understand how interactions between endothelial cells and matrix are impacting placental angiogenesis, we isolated endothelial cells, placental stromal fibroblasts, and we took villus samples from 6 severe FGR subjects, 6 appropriately grown preterm controls, and 6 full-term controls with uncomplicated pregnancies. And although we tried to match preterm and growth-restricted subjects within 10 days of each other. We unfortunately weren't able to do that. So you can see that the gestational age is significantly different among the three groups. Route of delivery was also different, with all preterm controls delivering spontaneously, and then birth weight percentiles were obviously significantly different, and notably all growth restricted pregnancies had persistent, absent, or reversed umbilical artery Dopplers. And so we first wanted to get a better idea of whether stromal composition differed in severe growth restriction. And our collaborators helped us develop a protocol where we're able to decellularize villus tissue and enrich the remaining fraction for extracellular matrix proteins for downstream mass spec. And so after filtering the results to a 1% false discovery rate at the peptide and protein levels, PLSDA showed that there were significant differences in ECM composition in growth restriction as compared to either preterm or term controls. And because there's some overlap between preterm controls and term control specimens, I want to tell myself that gestational age has less of an effect on matrix composition than placental phenotype, although I have no evidence to back that up yet, and it's something we still need to investigate in more detail. It'd be great to not always have to have two control groups. And anyway, while this data is probably reasonably representative of villostroma, it does include matrix from the entire gamish of cell types that make up placental villi, which really kind of limits our ability to ascertain differences specific to villostroma. And so to more specifically address what might be going on with villostroma, we developed a method of using our placental fibroblasts to generate cell-derived matrices, which I'll abbreviate CDM from here on out. And so as proof of principle, we stained cells with CSPG4, which is a fibroblast-specific protein that's typically not excreted in the absence of stimulation. And you can see that when fibroblasts are cultured on plastic, CSPG4 is intracytoplasmically located. After several days of matrix generation, before we get rid of the cells, you can still see CSPG4 kind of in the cellular distribution. And then once we decellularize it, it is gone. So suggesting that the fibroblasts are gone with a little bit of leftover nuclear debris. And then so to better characterize a matrix, we chose to stain for fibronectin, which again is one of the most abundant ECM proteins within placental stroma. And so in pre-decellularization conditions, fibronectin again appears to be more distributed in kind of an intracytoplasmic fashion. But then post-decellularization, after the cells are removed, you can still see matrix that's laid down there, both in fluorescence and grayscale imaging. And so to determine just at baseline whether there was a global difference in matrix deposition by either control or growth-restricted fibroblasts, we also just did simple bright field imaging, and the blue filter is just a mask that was applied just to make things easier to see. But you can see that there's no obvious difference between the two groups in terms of global matrix deposition at day 0, 7, or 14 after decellularization. So after we were able to consistently generate these placental-specific cell-derived matrices, we then wanted to investigate how tissue-specific matrix impacted endothelial cell angiogenic properties. And so again, we isolated endothelial cells and fibroblasts from control and growth-restricted placentas. And then we took the fibroblasts and generated control or severely growth-restricted matrix. And then we compared 4 groups. So control cells on control matrix, growth-restricted cells on growth-restricted matrix, and then kind of a crisscross of the 2 control cells on growth-restricted matrix and growth-restricted cells on control matrix. That's always a mouthful. So the first thing we looked at was endothelial cell proliferation. And so control cells on control matrix, which is the solid black line, proliferated most robustly as expected. Not unexpectedly, the growth-restricted cells on growth-restricted matrix, which, I'm sorry, my arrow keeps coming and going, is the red dashed line, that these cells didn't proliferate as well. But what really caught our attention was that growth-restricted cells on control matrix, which is the red solid line, exhibited partial improvement in migration, while control cells on growth-restricted matrix, which is this black dashed line, showed the worst proliferative capacity, although technically, this wasn't significantly different from growth-restricted cells on growth-restricted matrix on post-hoc analysis. And so we also looked at cell migration via live cell imaging, which we think was the most striking finding. And so the left image shows spider plots, which essentially place all the origin of all cells kind of at the center of the graph. And then it shows how hundreds of endothelial cells migrate from that sort of artificially designated origin. And so for control cells on control matrix, you can see that cells moved quite well, leading to this expansive spider plot showing appropriate cell migration. And in contrast, these growth-restricted cells on growth-restricted matrix form this very tight spider plot. And when you actually track the trajectory of these cells individually with live cell imaging, the growth-restricted cells are moving, but instead of moving in a straight line, they look like they're moving more in little circles, in contrast to the control cells on control matrix. What's especially compelling again is that control cells on growth-restricted matrix didn't move so well, but growth-restricted cells on control matrix looked to have this significant rescue of their impaired endothelial cell properties. And then this last finding here was especially compelling to us because if you remember the tube formation images I had shown earlier, those growth-restricted cells were unable to be rescued by Matrigel. And so that suggested to us that there was something specific about control placental matrix or something more tissue-specific that could lead to enhancement of growth-restricted endothelial cell migration. And so to better get an idea of what was going on in our cell-derived matrices, we started out by looking at collagen-1 and fibronectin, and this was totally targeted solely based because these were two abundant ECM proteins. And in all three of our groups in terms of mass spec as well. And so here you can see that not only is there less expression of both collagen-1 and fibronectin in growth-restricted matrix, but the topography was also clearly disrupted in growth restriction. And while the bright field images that I had shown you earlier with the blue mask suggested that there was no difference in overall topography between the two groups, these images made us wonder whether bright field just wasn't sensitive enough to capture the nuances of CDM or whether growth-restricted fibroblasts were unable to uniformly deposit specific ECM proteins. And so to test this hypothesis, we stained for thrombospontin 1, and we chose this protein for two reasons. First, unlike collagens and fibronectin, which were abundantly expressed in all three of our groups, term, preterm, and growth restriction, thrombospontin was one of the most highly expressed ECM proteins only in growth-restricted villus tissue, with substantially lower expression in both preterm and term controls. And second, there's also some biologic plausibility to increase thrombospontin in growth restriction because it's traditionally been considered an endogenous inhibitor of angiogenesis. And so unlike collagen-1 and fibronectin, the deposition of thrombospontin 1 appeared similar in both groups and arguably even more uniform than in growth-restricted matrix. And all of this together suggested that important differences in matrix aren't just limited to quantitative differences in composition, but that topographical differences may make some ECM proteins more accessible and others less accessible to endothelial cell surface receptors. And so we wanted to start investigating placental matrix in more detail, and we did this in a variety of ways that in retrospect, we should have probably done more sequentially rather than all at once, but hindsight is 20/20. So I think I have about 5 to 10 minutes left, so I'm going to skip a few slides, but the first thing is that there is a clinical fellow in the lab who took on the somewhat reductionist approach, but answered some low-hanging fruit that needed to be addressed. And she wanted to know whether individual substrates alone, exogenously plated on plastic, would rescue anything. And I think the overall take-home point, just to skip these slides, is that there was no one magic bullet of individual extracellular matrix, which we weren't necessarily surprised about. So at the same time, we were also generating placental CDM that were subject matched to those same mass spec specimens that we used for total villus tissue. And these 18 CDM samples generated in triplicate continued to show similar levels of distinct clustering on PLSDA. So this was the total villus tissue, and then this is the CDM tissue, with slightly more overlap between term and preterm controls again. But looking more closely at the specifics, we found that like total villus tissue, fibronectin, collagen 1, and collagen 3 were the most abundantly expressed stromal proteins in all 3 clinical cohorts. In total, there were 72 proteins, though, that were differentially expressed as determined by one-way ANOVA with a false discovery rate of 0.01. And the 4 with the highest fold change of differential regulation are shown here. So EDIL3, tenacin X, this is thrombospondin 1, and this is TNF super family member 4, TNF member super, TNF super fam, whatever, TNF SF4, it's a TNF super family member. And so although we had started out being most interested in thrombospondin, we're also starting to look at these other 3 ECM proteins, and we're collaborating with ECM microenvironment experts and bioengineers to investigate how these 4 proteins affect stromal stiffness, how their quantity and topography impact how extracellular matrices present ligand to endothelial cell surface receptors. So this brings me, so this slide is just to show that these were the ones that were most differentially expressed only in growth restriction and differentially expressed as compared to both control groups together. And so the third prong of investigation that we sort of simultaneously pursued was how endothelial cells and matrix interact. And based on very early preliminary data at the time, we landed on fibronectin and started with 2 main fibronectin binding integrins, alpha V beta 3 and alpha 5 beta 1, which are both expressed on fetoplacental endothelial cells. And so as a brief reminder, integrins are plasma membranes that when activated, they ultimately connect ECM ligands like fibronectin to intracellular focal adhesions and cytoskeleton. And then this regulates various cellular downstream processes, including proliferation and migration. And so to specifically tease out mechanisms related to these 2 integrins and their interactions with fibronectin, all of the subsequent data I'll show are controller growth restricted cells plated on exogenous fibronectin substrate, but this was done by Diane Gumina, who was a phenomenal grad student who was incredibly well trained by your own awesome Dr. Wynn, and then worked with me at Colorado after we lost Dr. Wynn to you guys. And so the first question she had was she just wanted to kind of know what the individual roles of alpha 5 beta 1 and alpha V beta 3 were. And so she took controlling growth restricted cells and treated them with IgG control or antibodies that block alpha 5 beta 1 or alpha V beta 3 activation. And when alpha 5 beta 1 was blocked, you can see that control migration in the blue lines and FGR migration in the red lines were significantly impaired to a very similar degree. Although again, inherent differences existed where growth restricted cells moved more slowly. In contrast, we were surprised to see that there was no effect on growth restricted cell migration when alpha V beta 3 activity was suppressed. And instead, we found that when alpha V beta 3 was blocked in control cells, the degree of impairment was similar to that of growth restricted cells at baseline, but then there was no additional impairment in growth restricted cell migration when alpha V beta 3 activity was inhibited, suggesting that it might not be being used effectively in growth restricted cells. And so with these functional differences in mind, the first obvious question was whether there was any sort of baseline difference in total endothelial cell expression. And in the interest of time, I'm just going to say no and move on. And so she then looked at various ways of how integrin function is regulated. And so the first one was through integrin activation. And she tested this hypothesis in a couple of ways. The first way, I'm trying to balance my time with slides. So when
You have time. Don't feel pressured. You have time to go through it.
Okay. So one way that integrins are regulated is through integrin activation. And so how that happens is that internal signals inside the cell activate the integrin from its closed bent conformation to this extended conformation, where it can then bind to extracellular matrix ligands like fibronectin. And so we had hypothesized that activation could be impaired. And so Diane actually modified this technique to actually obtain membrane integrin complexes. And so she plated cells, controller growth restricted cells on fibronectin ligand. And after cross-linking and removing cell bodies with sonication and several washes, she was able to just get adhesion complexes alone that could then be used for downstream analyses. And in the interest of time, I'm going to skip over the proof of concept slide. And just show you that at least through automated capillary immunoblotting, neither alpha V nor beta 3, when normalized to total protein, exhibited a significant difference in activation. And this was very similar for alpha 5, although unfortunately, for some weird reason, even though we had optimized the antibody, we weren't able to see beta 1 on this just capillary immunoblot. And so we wanted to see, well, maybe there is just not a ton of beta 1 even at the membrane. And so she wanted to reassess this another way. And so she used TIRF microscopy, so total internal reflection fluorescence microscopy. And so unlike routine epifluorescent, the laser hits the cell at a different angle, and that's able to capture a super fine region of interest. And in this particular case, just at the membrane. And so using a combination of antibodies directed against active versus total integrin forms, she was able to look at alpha 5 and beta 1. And you can see that there were no significant differences for both, confirming her alpha 5 findings on capillary immunoblot before, and then also showing no difference in beta 1. And there was, despite beta 1 being present at the membrane. And so, although there was no difference in integrin activation, one other way that integrins can be regulated are through focal adhesion complexes. And these can differ because after integrins bind to extracellular matrix ligand, they can recruit other receptors like other integrins, different ones, not even just the same ones as themselves, that bind to different ligands, or they can recruit receptor tyrosine kinases like VEGF receptor 2, resulting in these focal adhesion complex formations that activate various signal transduction cascades, and then that regulate actin cytoskeleton to promote proper motility. And so to test whether this was the case, she did this crazy experiment, and I'm only going to go over the salient portions today, but she plated cells on fibronectin-coated chamber slides, and after 18 hours generated a wound, and then 3 hours later fixed the wells and stained for these, 11 different conditions, and then used TIRF once again and took single and dual channel images, depending on the number of stains, and then sent all the data off to the focal adhesion analysis server that's based out of UNC. And essentially for this analysis, she looked at 8 regions of interest per subject, but this ultimately incorporated thousands of focal adhesions. So although she had 11 staining parameters, I'm just going to, again, focus on the ones that are important today. And so looking first at active alpha V beta 3, she found similar quantities of active alpha V beta 3 containing focal adhesions in both control and growth restricted cells. But in contrast, there was a significant increase in focal adhesions co-stained with both alpha V beta 3 and paxillin, which is a main component of focal adhesions that are thought to represent a more immature focal adhesion. And with regard to alpha 5, despite no difference in alpha 5 beta 1 activation, she did see more alpha 5 containing focal adhesions, but she also saw more alpha 5 containing adhesions that were co-stained with Zixin, which in contrast to paxillin is found in more mature and more stable focal adhesions. And so I think together the increase in alpha V beta 3 paxillin and alpha-5 zixin co-staining in growth-restricted cells suggests that there's potentially an imbalance in focal adhesion maturation that's integrin-specific in these growth-restricted cells, which may be just one mechanism that drives aberrant signaling of anchorage-dependent events and can impair the proper establishment of cell polarity. And so because we saw that this alteration in focal adhesion maturation that's integrin specific was present, we then wanted to investigate whether dynamic trafficking was altered in growth restricted endothelial cells. And so normally integrins have to be shuttled through a cellular treadmill in order for the cell to move properly. And so from the plasma membrane, this integrin on the rear end gets endocytosed. It goes into an early endosome, which then ends up going toward lysosomal degradation or recycling, where it gets shuttled back to the front of the endothelial cell. So that allows the rear end to pick up and the front end to move forward and plop down and attach, so it can kind of shuffle its way forward. And so, to be entirely honest, this was not the best way we wanted to do this, but we had some technical difficulties, and for the purposes of her thesis, this seemed to be adequate. But Diane did the same wound scratch experimental design and used confocal imaging this time to assess the number of early or late endosomal vesicles per cell. And not surprisingly to us, we saw significantly fewer vesicles per cell for both early and late endosomes and growth restriction, suggesting that there's altered intracellular trafficking dynamics in these growth restricted placentas. But again, honestly, we wanted to see this more via co-localization, which we were having difficulty with. And co-localization doesn't necessarily address dynamics. So although she's done, we're planning on looking at this either with proximity ligation assays or even kind of these biotinylation assays, where we can then track individual integrins as they shuttle through various cellular compartments. And so in summary, there are inherent endothelial cell and matrix defects in growth restricted cells, and these bidirectional interactions regulate placental endothelial cell angiogenic properties. And this is a crude and extrapolated schematic of what we think is happening overall, but we think that in control placentas, endothelial cell integrins alpha V beta 3 and alpha 5 beta 1 bind to normal extracellular matrix. And there's a proper balance in focal adhesion maturation where there's proportionate alpha V beta 3 paxillin and alpha 5 beta 1 zixin that then induces downstream signaling to the actin cytoskeleton, telling it to contract and release appropriately in order to achieve appropriate cell migration. At the same time, we think that these integrins are being properly recycled via early and late endosomes, which further facilitates forward cell propulsion. In growth restricted placentas, though, we think that alpha V beta 3 and alpha 5 beta 1 bind to disordered extracellular matrix. And although their degree of activation may be similar to control cells, we think that the focal adhesion maturation cycle is disrupted. So you have too much alpha V beta 3 paxillin leading to erroneous downstream signaling, and you have too much alpha 5 beta 1 zixin, which tethers the focal adhesions to actin, and then it prevents the actin from polymerizing and depolymerizing normally. And then at the same time, because there are fewer early and late endosomes, we think that there could be inadequate recycling of the integrins, and altogether leading migration to be messed up. And so I'm going to skip the future directions, but I think ultimately the question is whether the fetal placental vasculature is a modifiable component in severe growth restriction. And I think it's attractive in the sense that angiogenesis is normally ongoing throughout all of pregnancy, in contrast to the maternal uteroplacental circulation that gets established very early in pregnancy. And so would it be possible to promote angiogenesis in placentas that exhibit absent or reversed umbilical artery Dopplers? And truth be told, I'm not being greedy, and I'm not expecting to turn like 300-gram fetuses at 23 weeks into full-term chubby babies. But even if we could help gain just an additional week or two for these super early babies, we might still be able to improve outcomes. And so I apologize that this was a little longer than I thought it was going to be, but thank you so much for your time. I'm grateful to all the wonderful people I work with. Thank you, Virginia, for the invitation, and I'm happy to take any words of wisdom or questions.
Great, Emily, thank you so much.
Sorry, it's talking faster and faster.
That's great. the extracellular matrix part of the story, I think this is the first time I've gotten to hear that. And it's pretty profound that the endothelial cells, which you are showing, there are some intrinsic differences and you have different functional capabilities, that if they're placed on extracellular matrix from actual placentas and controls, you don't see as much of a delta. And that it really seems like it's the real driving phenotype is the extracellular matrix, which then gets me to the question of the extracellular matrix is coming from the fibroblasts. And do you think it's, what is the environment that's altering the fibroblasts deposition of the ECM? Is it hypoxia? Is it a nutrient thing? It feels a little bit like a chicken and an egg question, because if there's not good angiogenesis, you're not going to have as good oxygenation.
Yeah, no, you hit the nail on the head. So I submitted an R21 a few weeks ago that I'm sure will get triaged to try and answer that, because I think one of the biggest criticisms of the work we do is that when we get these cells at the end of gestation, are they representative of actually the mechanisms that lead to that bad phenotype of growth restriction? Or is it because they've been like stuck in this cruddy in utero environment for however long, and now it's like their manifestation of that? And so that hopefully this R21 will eventually get funded at some point. But I think we don't know. And so there's so much about placental stroma in general that is unknown. And so we're trying.
I think that's a very profound aspect of your work, is that we can't ignore the stroma, the fibroblasts, the other aspects that could be regulating the angiogenesis.
Yeah, we're trying to develop 3D models and then like subjecting it to them to various insults to see if we can kind of quantify that, qualify that. And then trying to work with bioengineers to work on some atomic force microscopy, which is totally outside of my wheelhouse, especially when they start bringing out mathematical formulas. But hopefully that'll help answer some of these questions. But I think it's still that chicken and the egg question that always ends up making me just want to retire sometimes.
Oh, please don't, Emily. You're on to something here.
We'll see.
Yeah. Great. Any other questions?
It's like it's fascinating talk, and I feel you brought in a wonderful dimension for all of us to think about, even those who don't do this kind of research, Emily. So thank you so much.
Thank you for the opportunity to be here.
All right. Thank you so much, Emily. Look out your window and enjoy the Rockies for me.
Yeah, I will. Take good care. See you soon.
Bye.